All right, part 2 of chemistry is focusing on organic chemistry, and we learned that organic chemistry involves atoms and molecules, not atoms, but molecules and compounds that have carbon and hydrogen in them. And there are 4 macromolecules. Remember, those are big molecules that we're going to focus on, and that's our carbohydrates, proteins, lipids, and nucleic acids. So the first one is carbohydrates. Think about the name carbohydrate. It has carbon and hydrogen in it, and also oxygen. So this is our main source of energy, and carbohydrates you can write out here. These are your sugars, okay? They have carbon, hydrogen, oxygen. They do mix with water. You can mix sugar with water pretty easily. And they're, so spoiler alert, lipids are also made of carbon, hydrogen, oxygen, but the ratios are different. So if you just know something has C, H, and O, you don't know if it's a carbohydrate or a lipid. But carbohydrates, the H's and the O's are a 2-to-1 ratio. Okay, so it's actually a 1 to 2 to 1 ratio. So there will always be twice or around twice as many hydrogens as oxygen. In lipids, you're going to have hydrogens and very few oxygens. Like this might say 80 and this might say 3. So there won't be a 2-to-1 ratio. So back to carbohydrates, they are a source of energy. This sugar, think about giving kids sugar and how they go crazy. There are some classifications of these carbs based on their size. So mono means 1, saccharine means sugar. So this means 1 sugar. They're simple sugars. And these are examples are glucose and fructose, also galactose. Disaccharides, the prefix di means 2. So this is a double sugar. Sucrose, which is your table sugar, what you put in your sweet tea. And lactose, which is found in milk. Sorry, I got distracted. Poly, many sugars. These are complex carbs, huge carbohydrates. Starch is an example. Starch molecules made of hundreds of glucose molecules. Sucrose is made of 1 glucose and 1 fructose. Lactose is made of 1 glucose and 1 galactose, that other monosaccharide. So 2 monos make a di, and then 50 to 100 make up a poly. So these are huge sugars. Starch, anything plant-based that's white is usually starchy. Glycogen is, so starch is what plants store their sugar as. That's why we steal it from them, like the potatoes. Glycogen is how animals store their sugar. And so this is going to be stored in your muscles and liver. And then cellulose is actually plant cell wall material. This is what we would call fiber. We can't digest this, so it helps you move things faster in the digestive system. So this is what glucose looks like. And you can write it a number of ways. So here's the 1, 2, 3, 4, 5, 6 carbons. It's going to have 12 hydrogens. Feel free to pause and count. And then 6 oxygens, that 1 to 2 to 1 ratio. But usually sugars aren't these straight chains of carbons. They're usually found in ringed structures. So 1, 2, 3, 4, 5, 6 carbs, 12 hydrogens, and 6 oxygens. And this is a lot to write. Organic chemists get tired of writing all the letters, and so they shorten it to this, where every corner here is a carbon, and then a carbon would be stuck up here. So this is a ring of sugar. And so that would be a monosaccharide. This is a disaccharide. Here's this bond between the 2 monosaccharides, and then this is a polysaccharide, many monosaccharides. Lipids. Now remember, lipids are also made of carbon, hydrogen, oxygen, but it's not going to be a 1 to 2 to 1 ratio, and I'll show you that in a second. Also, lipids are insoluble in water. They do not mix with water. So this is going to be fat, steroids, and phospholipids. Why are lipids so important? Well, phospholipids make up the cell membrane. That's huge. And steroids are, some of our hormones are steroids. We're going to find out that cholesterol helps with cellular function. And then fat has a lot of useful functions like storage of energy, insulation, protection, that kind of stuff. So most lipids are fats, and the science name for a fat is a tri-3, that means 3, glyceride. And so this is stored energy, and there is more energy in one triglyceride or 1 gram of triglycerides fats than carbohydrates. That's why oil lamps burn so well, wax candles burn so well. There's a lot of energy in fats. There is energy in sugar, but it's going to burn pretty quick and be done. So why is it called triglyceride? There's a glycerol molecule and 3 fatty acids, so they call it triglyceride. And I already told you about phospholipids that make up the cell membranes. They're similar to triglyceride. Just take one of those fatty acids off and replace it with a phosphate group, and you have a phospholipid. Now, fatty acids can be categorized as saturated or unsaturated. Saturated fatty acids only have single carbon bonds. They are solid at room temp. We usually see them come from animals. So like your hamburger fat, chicken fat, pork chop fat. They're solid at room temp, right? Think about when you cook your hamburger and you leave and you come back to your pan to wash it and it's solidified. Those are saturated fats. And the way they're able to become solid is they're straight chained, and I'll show you what that means. Check this out. Anything that starts with S is going to be a saturated fat. Single, straight chain, solid at room temp. Unsaturated fatty acids have at least 1 double bond in them, and therefore they can't stack up because they're not straight. They're kinked, and so they're going to stay liquid at room temp. So this is usually plants. Think about your oils. Canola oil, right? Corn oil. Vegetable oil. Peanut oil, coconut oil. These things are usually liquid at room temp. Now, fish oil is the only one animal that has unsaturated fats mainly, and that's because it lives in water and it needs its fat to be liquid, or the fish would just be a solid thing. So when I say straight change, this is what I'm talking about. This is just single bonds, and it's very straight, and look, it's surrounded by hydrogens, and it's going to be solid at room temp. Saturated, solid, surrounded, straight. Unsaturated is going to have at least 1 or 2, maybe more double bonds, and so the fatty acid is no longer straight. It's got a kink in it, and because of that, you can't stack a whole lot of them on top of each other, and so they're going to be liquid. Now here's a triglyceride. So this is our fatty acids we were just looking at. Look at this one. Saturated. What about this one? Saturated and unsaturated. See, it's not totally surrounded by hydrogens because of that double bond. So it has a funky shape. Now, how many do we have? We have 3. So this is a 1, 2, 3 triglyceride. This is fat. Remember that phospholipids are similar to triglycerides, except they only have 2 fatty acids and a phosphate group added, and they are what make up cell membranes. So you want to always think about phospholipids making up cell membranes. Now, steroids are 4 connected rings of carbon with very little oxygen, like most lipids, and we see them used all over the body. They are a component of cell membranes. They are used to make hormones, and cholesterol is the number 1 steroid for the body. So here's the triglyceride. They didn't write all those carbons and hydrogens out here, but this would be the triglyceride. Take one of those fatty acids off and replace it with a phosphate group, and now you have a phospholipid. And the way we draw that is this little structure. So you might have heard of a phosphate head and lipid tails. So call these lipid tails. These are the fatty acids, the 2 fatty acids, and this is the phosphate glycerol side. Now this is amphipathic, which means it's partly polar and partly nonpolar. So the fatty acids are nonpolar, and then this is the polar part. So this interacts with water very well, and this side doesn't. So here we have a general structure of a steroid, and there's a few different types of steroid. Most commonly, this is cholesterol. So I'm seeing these 4 rings. Everywhere that there's a corner is a carbon, unless told differently, and there's always hydrogens associated with carbons in organic molecules. Where are the oxygens in here? Oh, there's one. So see what I mean? The hydrogen and oxygen ratio isn't 2 to 1, it's 2 to very few. Okay, our next organic substance is proteins, and proteins are used for a lot of things. So I have you filling in quite a few blanks here. We're going to build stuff out of it, so it's structural. It can be used as an energy source. Now, the first thing you're going to burn is sugar. It's your primary energy source. Then you're going to burn fat. And if you don't have any fat left, you'll burn protein. And this is called starving. You don't want to burn your protein. But as a last resort, you can. Some hormones are made of proteins or the amino acid parts. Receptors on the membranes are made of proteins. Enzymes are proteins, and antibodies are proteins. So what are all proteins made out of? Amino acids. So what are all polysaccharides made out of? Monosaccharides. What are fat made out of? 3 fatty acids and a glycerol. What are proteins made out of? Amino acids. And you don't need to know about the carboxyl group and the amine group or the R group, but do know they are connected to each other with specific bonds called peptide bonds. These are fancy word for covalent bonds between 2 amino acids. So here's this amino acid, and so this is cysteine. Now this R group is all amino acids have this structure, and this is their variable group. It can change, and that changes the name of the amino acid. So this is cysteine. It's the only amino acid that has sulfur in it. So remember that trace element is found in our protein. And then this is phenylalanine, another type of amino acid. So these chains up here, these R groups can get quite crazy. And so when you take this amino acid and this amino acid, you can bond them together with a peptide bond. And then proteins have levels of structure and conformational shapes and denaturation. And I do not want to go into this. This is kind of beyond the scope of our course. So put an X on this slide and an X on that slide because it's showing you the picture of the protein. The only one thing I would say is look at all those little amino acids held together by peptide bonds, and amino acids build proteins. Our last organic molecule is the nucleic acids. And so you have DNA, which is the genetic code. What's unique about DNA is it can replicate itself. And then RNA is another nucleic acid. That's what the NA stands for. So this is deoxyribonucleic acid, and this is ribonucleic acid. They're both nucleic acids. This one is actually made from DNA. So DNA can make more DNA, or DNA can make RNA, and that RNA is usually used for protein synthesis. And they have a specific code, and you don't need to know that. But do know that nucleic acids are made of nucleotides. DNA is double-stranded of nucleotides, and RNA is single-stranded. So this is one nucleotide. It has a sugar, phosphate, and base. All this is carbon where these bonds are. So it is an organic molecule. And so here's RNA single-stranded, and here's DNA. It's double-stranded. And these are hydrogen bonding, actually, between hydrogens and nitrogens in the molecule. And so this is a huge polymer made of many nucleotides. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 nucleotides making up that tiny short strand of DNA. And here's RNA. The difference in DNA and RNA, besides that they're stranding, is RNA is called ribonucleic acid because it's made with the sugar called ribose, and DNA is made with deoxyribose. So to review that, DNA stores your genetic code. It has deoxyribose as a sugar. It is double-stranded. It's called the double helix. A helix is like a wavy structure, and it's made of nucleotides. RNA RNA is actually made from DNA and helps with protein synthesis. It has the sugar ribose. It is not a double strand, it's single, but it's also made of nucleotides. Nucleotides make up all nucleic acids, and we have two examples, DNA and RNA. And this is just nice to know, that a CT scan versus a PET scan. And so we're just kind of throwing in a little bit of technology used in the doctor's office. So a CT scan is to also look at internal anatomy, and it uses X-ray emitting device, and it creates like a 3D image of the soft tissues. And so this can help you look at different densities and see about tumors. And there's a positron emission tomography, which uses isotopes, we've heard of those, that emit positrons, which are weirdo electrons, don't worry about that, and that detects biochemical activity, so you can look at how the brain's functioning or blood flow. Okay guys, that is all for chapter 2, so you have a couple labs to do with chapter 2 and practice to help you practice the material.